Hi, this is Dr. Rosario, and welcome to our first video in our series on microbial metabolism. In the last module, we talked about microbial growth and nutrition. One of the requirements for microbial growth is energy. In this module, we are going to focus on the biological processes that microorganisms use to obtain energy, specifically to obtain energy from their food. The first goal of this video is to give you an introduction to metabolism. This overview will give you an idea of how all of the individual processes that make up metabolism work together to support microbial growth. We will define several terms including catabolic, anabolic, exergonic, and endergonic. These are all very unfamiliar words, and it will be important that you have them clear in your head as you proceed through the module. Next, we will discuss the role of enzymes in controlling metabolic reactions. And then finally in this video, we are going to discuss oxidation and reduction reactions. This video will lay the foundation for understanding metabolism. So it's very important that you understand the content in this video before you attempt to move on to the others in this module. As always, I'm here to help you. So if there's anything that is unclear or if you need an alternative explanation, don't hesitate to reach out to me through our Canvas course or just email me directly. I am here to help you. The term metabolism refers to all of the chemical reactions that happen within a cell. This image is a map of the metabolism of E. coli. Each dot on the map represents a different organic molecule, and the lines represent chemical reactions that are used to build or break down those molecules. As you can imagine, the study of metabolism can be quite complex. In this course, we're not going to go into deep detail. By the end of this module, you should have a basic understanding of how microorganisms are able to extract energy from their food. You will also have an idea of how we can use some of these metabolic characteristics to grow and identify microorganisms in the laboratory. So the term metabolism refers to all of the chemical reactions that happen in the cell. We can divide metabolism into two halves. Chemical reactions within cells can be grouped into two categories: catabolic reactions and anabolic reactions. Catabolic reactions break down large organic molecules into smaller components, like we see here in this picture. Energy that is stored within the chemical bonds of our large organic molecules is called chemical potential energy. Catabolic reactions release this energy and use it to make ATP. ATP is the energy currency of the cell. Because energy is released, these reactions are considered exergonic. Energy exits the process. The smaller molecules that are made may be recycled and incorporated into other molecules, or they may be released as waste product as a result of these catabolic reactions. Anabolic reactions are the opposite of catabolic reactions. Anabolic reactions are used to build larger, more complex molecules. They require an input of energy and a source of raw materials. Reactions that require energy to proceed are called endergonic. Right? Energy goes into the process. You may recall in the last module we discussed how photoautotrophs capture the energy of the sun and store that energy in the chemical bonds of sugars. These organisms make their own food. Chemoheterotrophs, on the other hand, consume other living things as a food source. The most common form of chemical potential energy in living things is glucose. Cells must be able to convert this energy into a form that is usable. That is where ATP comes in. ATP is called the energy currency of the cell because it can be used in many different ways to perform cellular work, from building biological molecules to transporting materials across the membrane, to moving cells from one place to another. Much of what we will talk about in this next video will be the steps taken by cells to convert the energy that is stored in glucose to ATP. ATP is an energy carrier molecule. It is composed of the sugar ribose, the nitrogenous base adenine, and 3 phosphate groups. The 3 phosphate functional groups attached to the molecule are held together by high energy bonds. Cells use energy from the sun or other chemicals to attach a phosphate group to adenosine diphosphate to make adenosine triphosphate. This is called phosphorylation. This is my favorite 5-syllable word, phosphorylation. Don't be afraid of these big words. Removal of a phosphate group from ATP is called dephosphorylation or ATP hydrolysis. This process releases energy that can be used directly to perform cellular work. ATP is at the center of metabolism. It serves as the bridge between our exergonic catabolic reactions and our endergonic anabolic reactions. The process of extracting energy from organic molecules like glucose involves redox reactions. Redox reactions occur when an electron from one molecule is removed and given to another molecule. So here, this electron is being moved from A to B. The process of removing an electron is called oxidation, and the process of adding electrons to a molecule is called reduction. Now you might be thinking, well, that doesn't make a lot of sense. We're adding something. Why are we calling it reduction? But it's called reduction because we're reducing the charge of that molecule, right? Electrons are negatively charged. By adding electrons onto B here, we've reduced its charge. We've made its charge more negative. That's where that term comes from. When one molecule is oxidized, another is always reduced. We're always moving electrons from one place to another. The molecule that loses electrons is called the electron donor, or sometimes you might hear it referred to as a reducing agent. The molecule that gains an electron is the electron acceptor. Right? And it's also sometimes called the oxidizing agent. While electrons can travel from the electron donor to the acceptor in a single step, in biological systems, redox reactions often happen in many sequential steps. In these cases, an intermediate molecule can serve as a temporary electron carrier. These molecules can help transfer the electrons from one molecule to another. One electron carrier that is commonly used by cells is nicotinamine adenine dinucleotide, or NAD. NAD is positively charged. When it receives electrons from an electron donor, it combines with a hydrogen ion to become NADH. NADH is the reduced form of NAD. It can bring electrons then to the electron acceptor. As it does so, it becomes oxidized and is ready to go back and pick up more electrons. So NADH is the reduced form and NAD is the oxidized form. And it serves to carry electrons from one set of reactions into the next, from one molecule to another. Now, I like to think of our electron carriers kind of like dump trucks, right? A dump truck simply carries loads from one place to another. like a construction site or something like that. And what happens if the dump trucks all get filled up, right? Then construction can't continue. That's the same thing with our redox reactions that happen within our cells. If the electron carriers can't get rid of their electrons, then metabolism can't continue, right? There has to be a flow of electrons away from those electron carriers to their final destination, to their what we call the final electron acceptor. So that's important to keep in mind as we go forward. So far, we have discussed catabolic reactions and anabolic reactions, ATP, and redox reactions. The last thing we need to talk about before getting into the details of metabolism is enzymes. Enzymes are biological catalysts. A catalyst is a substance that speeds up the rate of a chemical reaction. To understand their role in the cell, we need to talk a little bit more about the dynamics of chemical reactions. Earlier, when I was discussing catabolic and anabolic reactions, I defined our exergonic and endergonic reactions. Each reaction has energy associated with it. It either releases energy or needs an input of energy to proceed. If it releases energy, we call those spontaneous reactions. They're energetically favorable. Here we have an example of a chemical reaction where a disaccharide is broken into two monosaccharides. This would be an exergonic reaction where energy is released. We can plot the amount of energy of the reactants and the products on a graph. You can see that the reactants have more energy than the products, and the difference between the two is called delta G, or the change in free energy as the reaction proceeds. Delta G represents the amount of energy, in this case, that is released in the reaction. Activation energy is the amount of energy necessary to reach the transition state in a reaction. The transition state is a molecular conformation between the reactants and the products. So it's kind of in between the two molecules or sets of molecules. As the reaction proceeds, the reactants must travel through this transition state to become the products. In order to reach that transition state, there is going to be a little bit of energy necessary for this to occur, and this is our activation energy. Energy is released, however, as the molecules transition from the transition state to the products, right? So in an exergonic reaction, the amount of energy released moving from the transition state to the products is greater than the activation energy. It is important to keep in mind that activation energy controls the rate of our chemical reactions. If we trace the amount of energy as this reaction proceeds, you can see that there's initial input of energy to reach that transition state before energy is released to make products. So here we've got our reactants and our products, and then our transition state has a higher amount of energy. We can trace that energy pathway as we progress through the reaction, and that difference between the reactants and the transition state is our activation energy. Notice that the overall change in free energy here is not affected by activation energy. Activation energy, like I said, it doesn't determine whether or not a reaction is exergonic or endergonic. It determines the rate of a reaction. Most chemical reactions that occur in cells, even those that release a lot of energy, proceed too slowly to sustain life because of this need for activation energy, this push of energy to get started. This is where enzymes come in. Enzymes speed up reactions by lowering activation energy. Enzymes are usually proteins that are biological catalysts. They speed up the rate of reaction without being used up or permanently changed. They do not change the overall delta G, or amount of energy of a reaction, just the activation energy. They do this by bringing reactants together. Sometimes they distort the shape of those reactants. helping them get to the transition state. They might provide a microenvironment for reactions to occur in, and sometimes the enzymes will actually participate in the reaction, temporarily holding functional groups or atoms as they're being moved from one place to another. The image on the screen is an example of an enzyme, so this would be what's called a space-filling model. It's important to remember that enzymes have three-dimensional shape. Like all proteins, the shape of an enzyme determines its function. In an enzyme-catalyzed reaction, the reactants are called substrates. In this image, you can see the substrate. It's depicted by a molecular model in black, binding to the enzyme through non-covalent interactions. The place on the enzyme where the substrate binds, it's colored in blue and red here, that is called the active site. Again, the three-dimensional shape of these molecules, both the enzyme and the substrate, and their chemical properties are essential for this interaction to occur. Sometimes an enzyme needs an additional non-protein component to function. These non-protein components are usually called cofactors, or sometimes they are called coenzymes if they are complex organic molecules. An enzyme with its necessary cofactors is called a holoenzyme. The protein by itself is called an apoenzyme. Enzymes bind to substrates at the active site, forming an enzyme-substrate complex. They catalyze the chemical reaction and release the products. This process can occur over and over again very quickly, sometimes as fast as hundreds of reactions per second. I've stressed the importance of enzyme shape. Anything that can alter the shape of an enzyme can alter its ability to function. Because enzymes are generally proteins, temperature and pH can be an important factor. Most enzymes function best at body temperature, 37 degrees Celsius, and relatively neutral pH. But there is a fair amount of variation, and it depends on the environment that enzyme has adapted to and that organism has adapted to. In addition to the environment, there is a fair amount of interaction with other molecules. So other molecules can influence enzyme activity. These molecules can bind to enzymes and affect their shape, for example. So one type of molecule that can interact with an enzyme is called a competitive inhibitor. Competitive inhibitors are molecules that bind to the active site of an enzyme and block the ability of a substrate to bind. On the other hand, molecules that bind elsewhere on the enzyme and inhibit substrate binding are called non-competitive inhibitors. Binding of non-competitive inhibitors can, but not always, alter the shape of the active site, like we see in this image. Why do we care about enzyme inhibitors? Well, because inhibiting specific biochemical reactions can be a way to control microbial growth. For example, the first widely available antimicrobial drugs were sulfa drugs, and these are competitive inhibitors of the folic acid synthesis pathway in many bacteria. So today, scientists study specific enzymes in an effort to create designer drugs that may be able to overcome resistance to our current selection of drugs that we have. Antimicrobial resistance is becoming a bigger and bigger problem. So understanding specific enzymatic pathways and targeting those pathways may be a way for us to get around antimicrobial resistance. Okay, so in this lecture, I've just kind of given you an introduction to the basic concepts that you're going to need to understand so we can talk about metabolism in more detail in the next couple of lectures. It's really important for us to understand how these organisms are able to extract energy from their environment, because that's what powers their growth, right? And of course, when we're talking about health and disease, we want to control the growth of microorganisms, right? We want to prevent the growth of many microorganisms, those that are our pathogens.